And our first speaker is Alireza from Shushmita Roy's lab. Great.
Okay. Can you hear me? Okay. Thanks to the organizers for giving us the opportunity to present our work. Today I'm going to talk about incorporating noisy prior for estimating transcription factor activities and how we can use these estimated TF activities to infer better regulatory networks. Regulatory networks model how regulatory proteins can control the expression of target genes. For example, here we show two transcription factors, X1 and X2, that binds to the promoter of a target gene and induce its expression. We depict these regulatory programs using directed graphs like this. For example, here X1 and X2 are parents of target gene Y, and the goal of network inference is to find a genome-wide regulatory network for a given cell. There are different strategies for inferring regulatory networks. There are experimental methods like chip-chip or chip-seq or knockdown experiments, and there are also computational methods like finding sequence-specific motifs or expression-based network inference that we are going to look into in this talk. In the most simple case, expression-based network inference start by the expression of the regulators and targets and infer a regulatory program that can explain the observed expression. These methods can potentially infer a genome-wide regulatory network. However, usually there is low overlap between the inferred networks and experimentally validated physical interactions. There are two major paths for improving the performance of these expression-based network inference methods. There are methods that are incorporating additional constraint or additional information. For example, incorporating imposing modularity constraint on the inferred networks or incorporating prior knowledge about the interactions of the network. The other way that we are going to look into in this talk is relaxing the assumption that we should use the expression of the regulator as their activity. And there are methods that are using this path too. One of the methods that can be used to estimate TF activity is network component analysis, or NCA. And there have been methods that have been using this method to estimate TFA and improve the performance of the inferred networks. For example, this publication by Arieta Ortiz and colleagues that use TFA to infer better regulatory network for B subtypes. The way that the NCA works is that it, as input, it takes expression of the target genes and the network that connects the regulators to target genes and estimate a TF activity profile that can explain the observed expression. It assumes a linear model, which means that the expression of target genes would be a linear combination of TFA activity of the regulators as specified by the adjacency matrix. The way that it works is that it fixes the regression coefficients, estimates the TF activities, then fixes the TF activities, estimates the regression coefficients, and repeat until convergence. NCA starts with the input network, and the question that we were interested in was, what will happen if the input network is noisy? To answer that, we started a simulation. We start with true TFA and a true network. We simulate the expression of that, and then we add noise to the true network and use that to estimate TFA using this noisy network. Then we use the noisy network and estimated TFA to infer a better, a regulatory network. Then we can compare the estimated TFA to the true TFA and inferred network to the true network and see how they perform. What we see is that as we increase the noise, similarity of the estimated TFA to the true TFA decreases, and also the ability of the inferred networks from those estimated TFA to recover the true network decreases. To help to improve the performance, we add a regularization term to NCA. So what we do is that we add a penalty term like this when we are estimating the regression coefficients. And the way that it works is that by imposing a stronger sparsity, we can remove the interactions that we have lower confidence in. And one of the ways that we can do this is using something like modified elastic net that was used in the Greenfield et al. Paper. So this would be the pipeline of our method. We start with a noisy prior network. We use regularized NCA to estimate TFA. In the regularized NCA, we have a penalty term to remove the interactions that we have low confidence in, and then we can use the estimated TFA and the noisy prior to infer a better network. When we look at the estimated TFA from the regularized NCA, we see that their similarity to the true TFA increase as we increase the regularization. And we can also see that the networks that are inferred from these estimated TFA perform better when we are using the regularized NCA. Next, we applied our method to a compendium of expression data from yeast. We used expression data from these publications that have 377 different samples. And for prior, we used a motif-based prior network. To evaluate, we can look at different metrics like predictable TFs or functional edges, but here we only look at the area under precision recall curves. We compared our inferred networks to gold standards that are based on chip or based on knockout. Here, the dashed green line is what we expect to see by random chance, and the red dashed line is the performance of the prior network. When we only use expression of the regulators and no prior, we see that we perform worse than the prior network alone. When we incorporate the prior but not TFA, we perform slightly better than the prior network. When we incorporate the TFA, original NCA, we improve over that, and when we use the regularized TFA, we improve over that. So we can summarize this with area under PR curve. We are showing 4 different methods. Gini-tree and Merlin don't use prior, and Inferlator and Merlin P use prior. We see that when we are only using expression, methods that are using prior perform better than the methods that are not using prior. TFA improves the performance of all the methods, and regularized TFA improve over that. We see this for all the gold standards that we looked at. Diagonal plots are the gold standards, and off-diagonal plots are the overlap of 2 gold standards. We see that for the knockout-based gold standard, the improvement is smaller, but overall, for all the methods and all the gold standards, we see that regularized TFA improve the performance of the network inference. So next, we applied our method to expression data in mammalian cell lines. We looked at mouse embryonic stem cells, human embryonic stem cells, human LCL, and MCF7. We downloaded the expression data from publicly available sources like GEO, and we also search using meta-SRA, which allows us to search the metadata of the SRA database. For prior, we use the CSBP motif and also ENCODE and Jasper motifs. And to create the prior network, we use PIQ program that scores the motif instances by the footprint of the DNAs from the same cell line. To evaluate the inferred networks, we compare them to gold standards from different sources. For mESC and HESC, we have a chip-based gold standard and knockdown-based gold standard, and we also have a small literature-based gold standard that we curated from different publications. For LCL, we only have chip and knockdown, and for MCF7, we are only looking at chip. Overall, we downloaded samples from 691 different data sets. we processed around 10,000 different samples, and after collapsing the replicates, we end up with overall 4,700 samples. And we select somewhere between 4,000 to 6,000 genes for each of the cell lines to infer network. Here we are showing the performance of the our inferred networks for different gold standards in most ESC. What we see is that when we are using the prior, we perform better than not using prior. TFA always improve over not using TFA, and we see that in most cases, regularized TFA perform better than original TFA. We see this for all the gold standards, chip-based, knockdown-based, overlap of chip and knockdown, and also the literature-based gold standard, and we see this for networks inferred from RNA-seq data or array data. We see similar trends for the human cell lines. Each row corresponds to one cell line, each column corresponds to one type of the gold standard, and we see the same trend. In most cases, using prior improves the performance. In most cases, using TFA improves the performance, and usually regularized TFA perform better than original TFA. Now that we have the inferred networks, we want to use that to improve our understanding of the regulatory networks for mESC. Let's say we have an incomplete gold standard. If we have additional important regulators that is unknown to the original gold standard, we can use that to improve our understanding of the ES state. So to find the important regulators, we train a linear model for each target gene. Then we remove one of the edges and ask how much the predictive power of the model decrease after removing that. We can sum this decrease of predictive power for all the edges of one transcription factor. That means that if we remove the transcription factor from the whole network, how much the predictive power of the whole network would decrease. And we can use this as a metric for importance. When we look at the transcription factors sorted by their importance, we see that the transcription factors that are important in ES state tend to rank higher. So we can use this to improve our understanding of the regulatory network. We have our incomplete gold standard. We have the potentially important regulators that is novel, and then by knocking that down, we can see if that will affect the ES state or not. Sonny Grace from our group and Caleb from Sridharan lab are looking over the predicted regulators, and they are going to test the regulators that we find. So in conclusion, we see that we can use the regularized TFA to estimate better TFA profiles from the noisy networks. We see that this can improve the performance of the network inference for both yeast and mammalian cell lines. We can use the inferred networks to rank regulators based on their importance for different cell lines, and we are testing these predictions for mESC. I want to thank my advisor, Sushmita Roy, members of Roy Group and Sridharan Lab that are helping us with this project, our funding agencies, and Center for High Throughput Computing that help us with this performance, this experiment. Thank you.
Okay, we have time for a few questions. I think Rich has a question. You might need to turn on the mic.
Great talk. So I thought a little bit about this. I think that's an amazing result, but you're sort of regularizing the activities to zero, and I wonder sort of what you're thinking about what to try next in the sense that the activities might all be half on and half off. Like with regulatory networks, we sort of know that, or we believe that sparsity gives us a very good place to set the maximum for our prior, which is at zero, right? But what do you think in terms of, with future work? What are the key principles? Are you regularizing to an arbitrary value, or do you think that regularizing to zero is where you'll continue developing this?
Well, that's a good point. Right now, for selecting the regularization level, we kind of do a grid search. That is not the best way to do that. But we are looking into other ways to do that. So here I only showed a couple of the regularization terms that we use for the simulation, but we use different ways to incorporate the regularization, and we also use different parameters. So right now we are doing grid search, but we are hoping to find a better way to set that.
So at the last slide, you saw how you evaluate the importance of the yes, the previous one, the next one, sorry. Yeah, here. So this is like close to backward stepwise selection. You remove one and you evaluate how important is it, right? Can you do the same thing but just adding one and see how it changes? So you follow the opposite path, basically.
I think we can do that, but because of the way that we set up our experiments, so we do this in, we do the network inference in a stability selection. So we infer 100 different networks, we take the consensus, and then we take the top 100,000 edges or 50,000 edges for this experiment, and we do the cross-validation to get the importance.